Welcome to Jinnious Lab. Today we will cover IB Biology B1.2.6-12 Structure of Proteins.
Today we're going to cover B1.2.6 to 12 Structure of Proteins. As we mentioned in the previous unit, amino acids, the R group defines the type of amino acid. So to go back, R group is the variable group that determines amino acid properties, and there are different types of properties, mainly 4 of them: hydrophilic, hydrophobic, polar, and charged. Hydrophilic means water liking, so it means that this amino acid can come in contact with water and other fluids. So if the amino acid has a hydrophilic R group, then the amino acid would attract water molecules and it will likely be charged, but it can be acidic or basic. If the amino acid has a hydrophobic R group, meaning that it doesn't like being in contact with water, then it would repel H2O molecules and it will be nonpolar. A polar amino acid would have charged molecules that interact with water, which is why hydrophilic amino acids are polar. And charged amino acids can have either a positive or negative charge, but positively charged amino acids would have basic acidity, and negatively charged amino acids would be acidic. Here's the structure of amino acids. You don't have to know these complex structure. You don't have to know like really any of them. This is just for reference to show different types of amino acids with different properties. So protein has 4 levels of structures. SO students, you only have to know until primary, secondary, and tertiary, and HL students, you have to know about the quaternary structure. You have to know that all proteins contain primary, secondary, and tertiary structures, so this is the base option, like every protein has these, but only some have quaternary structures. The primary structure is the first step, and it is the number and sequence of amino acids, which is determined by gene sequences. The bonding that is used is covalent bonds, which is the same as peptide bonds. Like your teacher or like other textbooks can use these 2 words simultaneously, so you don't have to like be confused between them. It means practically the same thing here. And the covalent bond occurs between the amine and carboxyl group, if you remember from the condensation reaction diagram. In the secondary structure, an alpha helix and beta pleated sheet is formed. Here is the alpha helix and beta pleated sheets. As you can see here, this is the alpha helix in a swirl shape, and the beta pleated sheet is just basically really just a sheet shape. So it's very easy to tell apart in diagrams. The bond used is hydrogen bonds between the amine and carboxyl group. In the alpha helix, the hydrogen and oxygen bonds into a coil shape, and then it becomes a coil polypeptide. And in the beta pleated sheet, the hydrogen bond between parallel polypeptides. There are hydrogen bonds between parallel polypeptides, which provide mechanical stability. So as you can see here, there would be hydrogen bonds between these parallel polypeptides, which result in a flat sheeted structure. In the tertiary structure, this determines the 3D conformation of the polypeptides. So it's basically a combination of secondary structures. As you can see here, there are multiple polypeptides combined, the 3D folding of polypeptides. You would have to know how to identify alpha helixes and beta pleated sheets in protein diagrams, but it's very easy to do because alpha helixes are just very squiggly and swirled, and beta pleated sheets, they are often marked as arrows and they're just flat. So you have to know 4 different types of bonds that occur in tertiary structures: hydrogen, ionic, hydrophobic, and covalent. The first bond, hydrogen bond, occurs between polar R groups, and ionic bonds occurs between charged R groups. When an R group gains a proton, it becomes positively charged, and if it loses a proton, it will become negatively charged. Hydrophobic interactions occur between nonpolar R groups. This is easy to remember because as we talked about here, hydrophobic R groups are nonpolar. So it would also, so this hydrophobic interaction would also occur between nonpolar R groups. So you would know that that water is polar. And so water forms hydrogen bonds with polar R groups, and nonpolar region, also known as also like hydrophobic regions in R groups that cannot bind with H2O's atoms, cluster in the protein interior to to minimize their contact with H2O. So they would be sort of like a cream donut. Like the cream part would be the hydrophobic amino acids, and the dough part that comes in contact with the air and stuff would be hydrophilic amino acids. The last type of bond is the covalent bond, also known as the disulfide bond or disulfide bridges. This type of bond is a disulfide bond between cysteines. A cysteine is a type of amino acid containing sulfur. You don't really have to know more information outside of this. And this is only for HO, but quaternary structures is linking of multiple polypeptides. And there are something called conjugated proteins, which are proteins that have prosthetic groups that are not protein parts. We will talk about this later on, so we would move on for now. This is a summary of R group interactions. This is just a diagram. You don't have to know how to draw these interactions at all. So as you can see here, a hydrophobic interaction is occurring and hydrogen bonds between a hydrogen and oxygen atom. And here's the disulfide bridge between cysteines and an ionic bond between charged amino acids. So because proteins are made up of amino acids, the characteristic of the amino acids determines the characteristic of the protein. So there are different types of proteins. So hydrophilic and polar amino acids would form the exterior that comes in contact with fluid like blood and like extracellular fluid as well. And while the hydrophobic and nonpolar amino acids would form the interior that are protected from the fluids outside. The example of proteins consisting of both polar and nonpolar amino acids are globular proteins and integral proteins. Integral proteins can include many proteins, and one of them is channel proteins. You would learn about this in later units. The thing you have to know is that hydrophilic and hydrophobic molecules only interact with each other. So hydrophobic and hydrophobic is fine, hydrophilic and hydrophilic is fine, but hydrophilic and hydrophobic molecules do not interact with each other. It just doesn't work. So there are different types of proteins depending on its forms. You have to know about fibrous and globular proteins in particular and their examples. Fibrous proteins are water insoluble. They're like elongated proteins. Just think of like sort of string fiber. They're long and they're made of repeated structures like sweaters. They usually provide structural support, strength, and stability in tissues, like in skin and tendons. And an example is collagen. Globular proteins are spherical and water soluble, and there are proteins with irregular folding. An example is insulin. So there are conjugated proteins. And the example you have to know about is the hemoglobin, which is made up of just normal proteins and a heme group. We will talk more about this here. So more specifically, the fibrous protein has 3 polypeptides twisted in a triple helix shape. And the twisted chain is maintained by hydroxyproline residues and polypeptides. You probably don't have to know this name, but it would be useful in long answer questions. The interactions included is hydrogen bonds and van der Waals force. Van der Waals force is very weak intermolecular forces between molecules, so I think it would be okay to just write hydrogen bonds. As you can see here, multiple protein fibers are twisted together to form a collagen fiber. Insulin is a type of globular protein, and insulin is a hormone that lowers blood glucose levels, and it's in non-conjugated proteins. So there are only two polypeptides, one alpha helix and one beta pleated sheet. There are hydrogen bonds, disulfide bonds, and hydrophobic interactions occurring within the insulin. And it has a hydrophilic exterior and a hydrophobic interior. And this is because insulin has to travel through blood because it's a hormone that is transported throughout blood. So the hydrophobic exterior allows insulin to come in contact with bodily fluids, and the hydrophobic interior stabilizes the insulin's round structure. As I mentioned briefly, hemoglobin is a type of conjugated protein. It has a heme group, which is a prosthetic group because it's not actually a protein. As you can see here, there are two alpha helices and two beta pleated sheets, and the heme groups, these purple ones. The heme groups bind with oxygens, and this is why the hemoglobin is responsible for transporting oxygen throughout your body. It's possible because the heme group allows the molecule to bind with oxygen and move in blood. There are different types of protein examples and its functions. You probably only have to know about like 4 or 5 of these. You don't have to memorize all of them. I will just briefly go over the names. Immunoglobulins, also known as antibodies, its function is defense against pathogens. Rubisco is an enzyme that catalyzes photosynthesis. You don't have to know this full name just yet. Insulin is a hormone that regulates blood glucose levels. Collagen is a structural protein that maintains skin and skin structure. It's rope-like. And spider silk, just spider. It composes spider webs. And rhodopsin is a light receptor pigment in your retina cells that allows you to see color. And hemoglobins are transport proteins in your blood that carries oxygen. And histone proteins are proteins related to DNA, which like sort of coils the DNA together in a tight coil. You will learn about this later. And actin is a contractile protein in your muscles that allows you to move your muscles. And albumin is an osmoregulator protein in egg whites and other substances like blood. But you don't really have to know the name, I believe. You only have to know this name for when you talk about albumin and egg whites and in relation to denaturation of proteins.
For IB45 Genius Lab, don't forget to subscribe and like.